Let's now react two amino acids together, two monomers, to make a dipeptide, which is a dimer. Maybe by now you've already realized that the term, little part di over here refers to two. So I'm simply going to start out by drawing two amino acids, and you have to be able to do this as well. So I'm gonna, with a carbon over here, there's another carbon over here, a double bonded oxygen over here, an OH group over here. If you want, you can count the number of bonds. There should always be 4 bonds going away from a carbon. And a hydrogen over here, a hydrogen over here. And over here, this carbon here has an R group, and the R is a rest, so this could be almost anything. And over here I have a hydrogen, and this is an amino acid, because this one here, the NH2, the 2H is connected to the N, that's the amine group. And this C, double bonded O, OH, that's the carboxylic acid group. So we have an amino acid, because it carries these two so-called functional groups. And that's a monomer, okay? A single unit. And what I want to do now is I want to connect a second one to that to make a so-called dipeptide. And it's a condensation reaction, so I should be forming some water. So what I'm going to do now is I'm simply going to draw over here. I'm going to draw the same thing. I'm going to draw the nitrogen over here. So I'm drawing the amine group right now. I'm just copying it. O, O, H. I have another R group over here. It could be a different thing than the amino acid on the left. So, and that's basically yeah, 2 monomers, and I need to react them together because I want to make a dipeptide and later on a polypeptide and a protein. So how do I do that? Well, the cell does the following. The cell will actually look at this OH group over here, and it will take this H over here. The bonds that you see over here between the carbon and the oxygen is broken. Also, the bond over here between this nitrogen and this hydrogen down here is kind of broken. And this, what we have over here, OHH, this forms a new bond, and we have H2O, and this is water. So we're essentially taking away water, and I always draw it like this, H2O. And the carbon over here and the nitrogen over here, it forms a new bond. And this bond here that we have, did it just erase that? Okay, that's also interesting. I didn't know that it's actually able to erase, the laser point is able to erase it. So essentially this one over here is now able, it has a name, and the name of this bond is referred to as a peptide bond. But the drawing here is not really nice, okay? Because the bond looks so large, and there's still the H2O in here and so on. So what I'm going to do is I'm simply going to copy the whole thing again, and I'm going to simply draw the dipeptide nicely to actually show you how it should look in real life. NC CO N H C CO OH. Over here I have the R, I have the hydrogen over here I have the other R, and over here I have the hydrogen over here, and then 2 more hydrogens over here. So that is basically a dipeptide. It is an example of a dimer. Okay? So, of course, not all dimers are dipeptides. It's clear. Dimer is simply a general description of a substance which contains 2 monomers. And if you basically are given something like this, then they might ask you identify the peptide bond. And then you basically have the following situation. You know, aha, the peptide bond is between a carbon and a nitrogen. Okay, but between which carbon and nitrogen? Between this carbon and this nitrogen over here, or between this carbon and this nitrogen over here? It's always between the carbon and the nitrogen, where the carbon has the double bonded oxygen. So this thing here, this is the peptide bond. And yeah, that is, so I'm gonna make a big arrow. That's the peptide bond. So I've basically, now you know why I have redrawn it, because over here at the top it's so clear because the peptide bond is such a big line. But in reality, it's not such a big line, and the size of the bond over here is drawn the same as the other bonds, so you might have a more difficult time actually identifying it. So for this reason, I have redrawn the chemical structure over here. Okay, so to summarize, a peptide bond forms between an amine and a carboxylic acid functional group. So we have the amine group over here, the NH2, and the acid, the carboxylic acid over here. And you basically remove water by a condensation reaction, and then you have a dipeptide. And that's basically, that's basically it. And if you want to add another amino acid, you can always do that, because you add another one over here. Yeah, and so on and so on, until you have a chain of amino acids, which is a polypeptide. So, and last but not least, if you join two amino acids together to make one water molecule, how many water molecules do you make when you join 200 amino acids together? And many people instinctively say, well, 2 amino acids makes 1 water molecule, and 200 amino acids makes 100 water molecules. But this is wrong. It's 199 water molecules that you make. It's always one less, because you form, when you connect 200 amino acids together, you must have 199 bonds. And for each bond, each peptide bond, you make 1 molecule of water. So that's it.